Today we are discussing about the topic desmosomes. Desmosomes, the word desmosomes is a Greek word. The term desmo means bond or fastening. Soma means the body. Desmosomes are a type of anchoring junction in an animal tissue that connect adjacent cells. The simplest function of desmosome is to adhere cells together. And the desmosome is an adhesive intracellular junction that is crucial to tissues that experience mechanical stress such as the myocardium, bladder, gastrointestinal mucosa and skin. Desmosome was first observed in the spinous layer of epidermis by the Italian pathologist Giulio Bisosero. This is the simplest structure of desmosomes, and another epidermal attachment complexes is adherence junction, tight junction and gap junction, and three types of gene families is present. Desmosomal catherines. Desmosomal catherines includes desmoglins and desmocolins, and armadillo proteins includes placoglobins and placophilins, and plaquins include desmoplakin. Desmoglins and desmocolins are the two types of desmosomal catherines. This is the brief structure of desmosomal catherines. In this picture, the desmoglin and desmocolin are the member of catherine superfamily. Cytoplasmic components of the desmosome associated with the desmosomal catherine tail through a series of protein interactions, which serves to recruit intermediate filaments to site of desmosome assembly. These desmosome plaque components include placoglobin and placophilins. The placoglobin and placophilins are the armadillo proteins. Linkage to the cytoskeleton is mediated by the intermediate filament binding proteins. Intermediate binding protein is desmoplakin, which associated with the both placoglobin and placophilin. Although desmosomes are critical for maintaining stable cell-cell adhesion, they are also in dynamic structure that contributes to cellular process beyond that of cell adhesion. Extracellular domain of the desmoglin and desmocolins mediate adhesion, whereas the cytoplasmic tails of these catherines associated with the desmosomal plaque proteins. The outer dense plaque consists of cytoplasmic tails of the desmosomal catherines, which binds to member of armadillo and plaquing family. The placoglobin, a member of armadillo family, binds directly to the cytoplasmic tails of both the desmoglin and desmocolins. And the desmoplakin, a member of plaquing family, interact with both placoglobin and placophilins. The placophilins, finally, the interaction between desmoplakin and the keratin filaments form the inner dense plaque and tethering the cytoskeletal network to the adhesion. And next, the desmosomal catherines. The desmosomal catherines are two desmoglins and desmocolins. Desmoglins are denoted as DSGS, and desmocolins is DSCS. These are the two types of desmosomal catherins. In humans, there are three isoforms of desmocolins and four isoforms of desmoclins. Role of desmosomal catherins in epithelial differentiation is the desmosomal catherins show complex development and a differentiation pattern of expression. The DSG2 means desmoclins 2 and DSC2 desmocolins 2 are expressed widely and in all desmosomal bearing tissues, whereas the other desmosomal catherins are predominantly expressed in stratifying epithelia. In the epidermis, all 7 desmosomal catherins are present, and DSG2, desmoclins 2 and desmoclins 3 are mainly distributed throughout the lower layer of the epidermis, whereas desmoclins 1 is expressed at a higher level in the upper layer. Desmoclins 4 is primarily expressed in the hair follicle and in the granular layer with respect to the desmocolins. Desmocolins 2 and desmocolins 3 are present in the basal and spinous layer, whereas desmocolins 1 is expressed in the granular layer. The differential expression pattern of the desmosomal catherins implies that desmosomes within different tissues are biochemically and functionally distinct. And these are the desmosomal catherins diseases. In haploinsufficiency of DSG1 leads to the palmoplantar keratoderma and other diseases, pemphigus foliaceus, pemphigus vulgaris, paraneoplastic pemphigus. These are the desmosomal deficiency diseases. And next, the armadillo family of proteins. Previously we talked about the placoglobin and the placophilins. These two are the armadillo family of proteins. The cytoplasmic association with the catherins are partially mediated by the armadillo family proteins, placoglobin and the placophilins. Placoglobin is the best characterized armadillo protein in the desmosome. Structurally, the proteins contains 12 arm repeats amino acids are present, and this is the structural representation of armadillo family of proteins. The central armadillo domain of placoglobin also interact with the desmoplankton. Placoglobin associated with the desmosomal catherins and the desmoplankton intermediate filament complex simultaneously. Placoglobin clearly plays a key role in desmosome assembly as revealed by a fifth human disease as mouse genetic model system. And next is placophilins. The other armadillo members represent in the desmosome is placophilins. Based on the number and organization of the central armadillo repeat domain, placophilins are predominantly found at desmosome instead of adherens junction. Both placophilins 1 and 2 exist as two isoforms. Two isoforms are existing placophilins. Placophilins 1 and 2. Based on structural analysis, the placophilins contain 9 arm repeat domains. The amino-terminal head domain of placophilin 1 associated with the DSG1 and desmoplakin and keratin intermediate filaments. The binding of the plakophilin 1 health domain to desmoplakin appears particularly robust, and this binding event drives desmoplakin recruitment to cell-cell junction. And, last is desmoplakin. Desmoplakin is the most abundant component of the desmosome and serves as the key linker between intermediate filament and the plasma membrane. Previously, we talked about the desmoplakin function.